Hi, everyone. Thanks for joining today. My name is Jason Hamlin, and I'm going to talk about how you can improve cell adherence in vitro using DendroTEK dPGA substrate. We'll walk through where adherence tends to fail in real-world applications, and I'll show how dPGA stands up to PLO, PDL, and PEI in long-term culture and usability. Here's a bit of background on myself and DendroTEK. So I'm Jason Hamlin. My background is a master's in microbiology from the University of Manitoba and a PhD in neuroscience from McGill, where I studied intracellular trafficking. I did an industrial postdoc at Sanofi, and after that spent several years in product management at Stem Cell Technologies, bringing advanced cell culture tools to market. I'm now the fractional chief product officer at DendroTEK Biosciences. DendroTEK is a small biotech founded in 2022 in Montreal, Quebec. We develop IP and manufacture reagents. And our key technology is a dendritic polymer called dPGA. Today, I'll talk about where this reagent fits into modern workflows and what users are seeing when they work with it. So first, we're going to talk about adherent cell culture workflow and exactly where we fit in. So here's a simplified view of a typical workflow. You have been doing a lot of work to create your cell line. Perhaps you've obtained patient lines, gene edited lines, and you're scaling them up. And you may even be doing a little bit of early differentiation or manipulation. And after you've scaled up enough, you move into the screen. Now, at that stage, cells are replated into high content formats, 96, 384, or 1536 well plates on whatever substrate you're using, and maybe some specialized media. You apply conditions and dose response curves, and then incubate for a prolonged period. You can perform live imaging or fix and stain phenotypic profiles for ICC or enzymatic readouts. Cells cultured for extended periods, obviously, are subject to a number of risks. So, obviously there's, genetic risk, drift or chromosomal aberrations accumulating over time. Of course, you have contamination risk, growth rate issues. Sometimes cells grow too slow or sometimes too fast. There's material availability and lots of other things, but for adherent cell culture, there's specifically detachment risk, right? Cellular lifting or peeling. So let's look at the factors that affect adherence. At the base, you have your cultureware. That's glass or your tissue culture treated plate. On top of that, most users apply a cationic matrix, such as PDL, PLO, or PEI, or in this case, dPGA. This positively charged layer provides an initial electrostatic pull that helps bring the negatively charged cell membrane down towards the substrate. Above that are ECM proteins like laminin, collagen, fibronectin, added either as purified components or as components of gels like Matrigel or Geltrex. Above that, you have the integrins bridging the cell membrane to the ECM. These are essential, but remember, they're disrupted during passaging with proteases or trypsinization, so adhesion during replating is not trivial. It's a multi-layered problem, and we're going to focus in on that cationic layer, where the matrix sits right on the tissue culture treated glass or plastic. So let's look at this example figure, the differentiation of iPSC-derived dopaminergic neurons over 52 days. On day 11, this is a normal and intact monolayer. On day 22, differentiation is progressing. You can see the neurites forming, and the monolayer is still mostly intact. At day 30, we see clumping emerging, and regions of the plate are now devoid of cells. And by day 52, the cells appear almost entirely as spherical clumps connected by vesiculated processes. So this is an actual differentiation protocol, and it illustrates exactly why long-term detachment looks like in practice. And I know many of us have seen this before. We've seen it before because it's extremely common. If you look through publicly available images, vendor protocols, academic papers, and product ads, you can see this clumping everywhere. Now, clumping or 3D culture is, of course, fine if you intend to do that. But for the purposes of high content imaging, microfluidics, MEA, or anything else requiring a consistent 2D monolayer, this becomes a major failure mode. And it ruins experiments. So in the kinds of adherent failures, we see clumping. Sometimes there's peeling and delamination. Sometimes there's floating cells, and neurite bundling and fasciculation. And there's a number of mitigations. We can adjust the substrate. We can play with the cell density. We can change our technique. And many people have tried a lot of things to mitigate this. So what we want to talk about now is DendroTEK dPGA. It's designed to fully replace PLO, PDL, or PEI in that cationic base layer that I showed in the schematic. And we think that this is a solution for that that clumping and peeling. But before we talk about show the data, what exactly is DendroTEK dPGA? Well, it's a 550 kilodalton dendritic polyglycerol with peripheral amine groups. And we'll get to the structure in a little bit. It's provided as a sterile, ready-to-use concentrated stock solution. It has a water-like viscosity, soluble in water and PBS, so it's really easy to work with. It's easy to dilute to the working concentration. You can store your working concentration at 4 degrees for up to 4 weeks, or your stock solution can be stable for up to 2 years. What does it do? Well, it substantially increases cell adherence, reducing the detachment of cells, completely replaces basically PLL, PLO, or PEI as a coating substrate. So how does dPGA perform? On the left panel here, we're looking at iPSC-derived neurons that were plated on PDL or dPGA. The green staining is for beta tubulin, and the blue staining is a nuclear counterstain. And you can see the clump on the left and basically the absence of clump on the right. Now, this is sort of more of a zoomed in picture, but we'll see this time and time again where users observe the reduction and better adherence in the dPGA condition. On the right side, we see iPSC-derived motor neurons with matrigel alone on the top, or matrigel plus a dPGA base layer at 50 micrograms per mil. And again, we can see the reduction in the size and number of clumps, and we get sort of a more stable and adherent monolayer at the bottom. So this is really one of the clearest demonstrations that dPGA is doing something and that it's helping that adherence. Now, dPGA also maintains normal neural activity. So if you look at the left figure, this is a current clamp trace. Both PDL and dPGA conditions show normal firing patterns. So that enhanced adhesion isn't impairing the physiology. And on the right side, we see iPSC-derived motor neurons that are plated on matrigel only or matrigel plus dPGA, as in the previous panel. Now, on the left, you can see some electrodes aren't covered. There's a lot of clumps. And on the right, you can see a more stable monolayer of cells, and all of the electrodes are covered, and all of the electrodes are active in that work. So we think MEA users are going to see a lot of benefit from testing out dPGA. Now, here's another example. dPGA supports neurogenin-derived neural cell culture. So in this experiment, the user took iPSC-derived neurons and plated them on PLO or plated them on dPGA. And what we can see is that the NGN2 neurons adhere more uniformly on dPGA compared to PLO. Then it supported a more stable monolayer with fewer clumps. The experiments on this work were done with dPGA after this initial observations. dPGA also supports co-culture applications. This figure shows a triple culture, neurons, astrocytes, and microglia. On the panel, we can see astrocytes are labeled in green, GFAP, the neurons in red in pan-neurofilament, and the microglia with IBA1. And we can see that beautiful merge. And we know that a lot of people are doing neuroinflammation co-culture experiments, mixing gene-edited astrocytes with wild-type neurons, or ablating, or changing to try to dissect and interrogate exactly the root causes of or cell autonomous or non-cell autonomous causes of complex diseases, which of course have interactions between a lot of different cell types. And these experiments can take weeks to establish. A lot of times you grow your astrocytes and neurons first, you grow your microglia up on the side, and then you mix them together, and it's only after several weeks do you obtain your test condition. And so having a failure is quite expensive and costly in time and resources. So we're happy to see that this user was able to publish successfully using dPGA for their triple culture experiment here. So to understand dPGA's performance, let's look at the molecule itself. Now, this schematic shows a simplified portion of dPGA. The real actual dendrimer is, in reality, is a lot bigger. It's too big to actually easily visualize. But you can just imagine a hyper-branched structure that creates really a complex molecule, a complex surface. And we're going to see that this actually creates an increased roughness on the cell surface, which might lead to the dPGA's performance. Now, if you look at the backbone, it's a stable ether backbone. It lacks peptide bonds, making the polymer inherently resistant to enzymatic cleavage. And the positively charged peripheral amine groups provide a high density of cationic interaction with the substrate. So these features, these structural features, actually support some of the performance of dPGA. And in fact, polyglycerols were originally explored as alternatives to PEG, to PEG, polyethylene glycol, because they're water soluble, non-toxic, and chemically stable. But with a little bit of modification, it seems like they could be very, very useful for cell culture applications that we're talking about today. So I mentioned that dendritic structure, and it in fact creates a rough surface. What we're looking at here is an atomic force microscopy map of a surface coated with dPGA. It shows that there's an increased roughness compared to PDL, which has a more smoother or topology. And we can see that it varies to 5 nanometers in height. And so this added nanoscale texture could provide more anchoring sites for neurites and cell surface proteins, perhaps more adhesion sites per unit of area. It's not clear how actually this nanoscale texture does affect the cell culture, but it's an interesting observation that happens with dPGA. On the next slide, this is a very interesting finding. We were able to get dPGA tested on an ultra high density MEA. This is a 240,000 microelectrode array built by Sony and available exclusively through Vitravo. And Vitravo was able to test dPGA plus laminin on the electrode, or PEI and laminin on the electrode. And they found basically consistently that PEI and dPGA worked at days in vitro 4, days in vitro 12 on primary neurons. I'll play the movie here, and you can see that the dPGA laminin supports normal activity, but the user noted preparing the solution was far easier than preparing the PEI. PEI has a high viscosity. It's very goopy, right? It's difficult to handle, and it can't be stored, so you have to make it fresh each time. The dPGA solution is easy to use and can be stored for a long time. So that was a cool finding, and please contact Vitravo directly if you want to learn more about their services and this very cool Ultra HD electrode. Now on the next slide here, we're going to look at some high content imaging workflow from the Center for High Throughput Chemical Biology at Simon Fraser University in Vancouver, Canada. The Center for High Throughput does screening assays. This is the group of Dave Vocadlo and Yanping Zhu, or Yan, in the group. Helped set up this experimental test with dPGA and laminin, or PLO and laminin, which is what they're normally using. And they immediately saw, again, what we see in what the other users see. There's an immediate reduction in clumping. They saw basically a nice uniform monolayer and reduced clumping in the dPGA laminin condition. Now, if you look at the top middle panel, that is a lyso-FQ assay. Basically, there's it's a substrate, a fluorescent substrate that's quenched, and when it's trafficked to the lysosome, it is cleaved, and then that's a direct measure for GCase activity, or glucocerebrosidase activity. And if you're familiar with GBA-associated Parkinson's, there's basically a linear almost relationship between the activity of GCase and the severity and onset of GBA-associated Parkinson's. So it's very crucial to understand small changes in GCase activity. And they are in the bottom middle panel. They took a patient line, or an isogenic corrected control of the patient line, and compared the lyso-FQ GBA activity. And interestingly, what they found is that they could see a significant difference between the dPGA laminin control and patient lines, whereas they couldn't see a difference between the control and patient line in their regular PLO laminin condition. So this takes basically a bunch of things. It saves them time. It allows them to study the patient lines and isogenic controls. And we're hoping to see more testing from the Center for High Throughput Biology. You can contact them at the htcb_admin@sfu.ca to learn more about their workflow. So, the factors in cell culture that affect adherence, there's several of them, and how do each of the conditions that we were looking at add up? We know PEI does have that high degradation resistance. That's why Vitravo is using it for that workflow. But as others have noted, the stability in solution, it's unstable. You need to make it fresh. dPGA is stable long term. We know that PLO is smoother. We don't have data right now for the surface nanotopology of PEI. We do know that dPGA has rougher surface. And the material cost for PLO and PEI is low. For dPGA, we'll call it medium, but it's cost comparable. I mean, you can get 5 milligrams for $80. And what we do know is the effort, the ease of use is quite difficult for PEI, but with dPGA, it's really easy to use. We do know that there's other substrates and treatments like plasma coating, the DAP treatment, We currently don't have data for this, but we're looking to test that alongside. So please stay tuned for that information. Now, I just want to wrap up with some user feedback. This first one was from SFU. Basically, they got more consistent outcomes in their imaging-based lysosomal enzyme assays. We have people talking about my stuff isn't peeling off the Maxwell chips. We have people mentioning that it's working for our cortical neurons. Again, people saying that they don't need the borate buffer or for PEI, and they're noting that in some cases it can even be cheaper that if it's used at the appropriate or optimized concentration. So we're getting a lot of this great feedback from our users, and we'll keep seeking out that feedback as we learn and grow in the cell culture market space. So in summary, DendroTEK dPGA provides stronger, more stable adhesion across long-term cell culture. It reduces clumping and peeling, enabling more consistent data. And it's very easy to use. It's affordable and a reliable coating that outperforms PLO, PLL, or PEI. That's the end of the presentation. To learn more, please email info@dendrotek.ca or visit www.dendrotek.ca. We're happy to answer any questions. Thank you.